well or not? I'm not entirely
Let's see. So you're in the middle. You must be in the middle. And Amit must be in the middle.
Just tell me where I go.
You want to put Laura in the middle, maybe? And I'll do the president.
Thank you. Okay. Yes, it takes two. Okay. So, as Vice Chair for Research, I am really pleased to welcome you to today's Grand Rounds, which is a special Grand Rounds because it's the Glassman Memorial Lecture. And we do both the Glassman Memorial Lecture, which is going to be delivered by Dr. Scott Rauch, which is great, and also the Glassman Award to a trainee, which is to Dr. Amit. How do you say your last name, Amit? Lazaroff. Yes. So let me tell you about both. So in this audience, I'm sort of curious, how many people here remember Sandy Glassman? Just raise your hand. So that's wonderful. So lots of people remember Sandy Glassman, and we were just speaking with his daughter about what a character Sandy was and a real innovator and creator and a longtime member of our department who Tim is reminding me actually hired Steve Roos and Tim Walsh. So that's, really they were his. And he unfortunately died in 2011, and with a very generous gift from the family. And this annual memorial lecture was set up. And there's a committee, and that's who you saw on the stage, which is Tim Walsh, Evelyn Attia, and Steve Roos, who knew Sandy very well. And each year they choose someone to, outstanding researcher in the field who's related to Sandy's topic of interest. And they chose Dr. Scott Rauch, who's a wonderful choice, and I'll talk about Scott more in a minute. In addition, besides the lecture, they also funded an award to a trainee that each year is given, and it actually comes with money, so it's not just a plaque. So Amit, open your envelope. And that's to honor research accomplishments during the residency or the fellowship. So any trainees in the audience, this is to alert you to be ready to apply for this next year if you want it. And I'm going to first call up Amit back up to the stage, and so we can do the official award of your plaque, your certificate, and your check. And Amit won this for his paper entitled Hippocampus Resting State Connectivity in PTSD. So congratulations. Yeah.
Congratulations. You have a certificate. You have a check.
Okay. So that's great. So now let me tell you about Scott Rauch, who's our lecturer today. So Scott has many titles. He's the president and psychiatrist in chief at McLean Hospital, and he's professor of psychiatry at Harvard Medical School and chair of Partners Healthcare, which is the massive, healthcare system up in the Harvard system, which actually we have envy about because it seems like an integrated system, unlike some other systems that we know. Scott, I believe, was born in the Midwest. And so, is that right? Oh, I'm sorry, Western Massachusetts. I'm sorry. He was an undergraduate at Amherst, where he was a neuroscience major. And that must have been one of the earliest neuroscience majors, I think. That was really, Amherst was ahead of its time. He then did medical school at University of Cincinnati. Then he came back to the East and did residency in psychiatry at MGH, and he also did a radiology research fellowship at MGH, and then did fellowship training as clinical investigator fellowship at Harvard and MIT. He then rose through the ranks at MGH and through the Harvard system, and as you see, has reached the professor and head of this and head of that in that system. So he, obviously was wildly successful. He's, of course, won many awards, has many publications, and has been an enormously smart and accomplished scientist. And I'm not going to describe to you what he does because he's going to do a better job than I, and he's going to talk to you today about that. But I am going to tell you things that he might not tell you about. One is, and I can embarrass you, Scott, I think Scott is really credited with really rescuing McLean. At the time that Scott went to go be president of McLean, I got told McLean was closing, the doors were closing, but no, the doors are not closing at McLean. In fact, Scott, I think you became head of McLean in 2006, is that right? McLean is flourishing with really high-level recruits, such as in my field, Carrie Resler and Suzanne Haber. Scott managed to pluck those two people to McLean over all the other places that they might have gone, as well as other things that he's done. So rescuing McLean is a huge thing that he's done. The second thing is he's mentored scores of scientists and rescued some of them from failure too. I have to think I wasn't rescued by you, but I was one of the people who was fortunate enough to be mentored by Scott, and Scott was one of my mentors on my K Award. And he's just a wonderful, thoughtful, strategic mentor. The way he rescued McLean, he really can see career trajectories for young people through science. The third is he's a really wonderful leader in a quiet, understated way where he really, his faculty at McLean tell me he's really created a culture of collaboration and transparency and integrity and merit. So the faculty really are incredibly loyal to him and what he's accomplished there. And finally, he's that word mensch, right? So he's not, people do wonderful things for their own research program or their own institution, but Scott actually does wonderful things for the field. So Scott's one of those people who's actually generative and generous outside his institution to people at other institutions. And that's really something that I admire enormously about him. He's just a good force in the field. And so I think the Glassman Committee made a fabulous choice because I think Sandy was good for the field in his own odd, eccentric way. And I think Sandy would recognize not only the science that Scott has done, but also his positive force. So it is my great privilege to honor Scott publicly in this forum, to invite him to the stage now, and he gets a plaque, and then he gets to speak to us. And the title of his talk is up there. So congratulations to Scott. Okay.
Thank you so much, Blair. That was very generous. It's been marvelous to see your ascent over the years and the fabulous leadership that that you're conducting here at Columbia. It's such a joy for me to come back here always. I was looking back in my records, and I've come back to speak here almost exactly once every 10 years through my entire career. And the one other little side note I'll say, which I shared with people at dinner last night, my grandfather graduated from the Columbia Pharmacy School here in 1924. So I'm thinking if you have me back again in about 7 to 10 years, we'll be able to, the Rauches will hit a century mark. And part of why that's not as tangential as it might otherwise be is I think, given that this is the Glassman lecture, it makes it all the more special, realizing what a fabulous, iconic figure Sandy was. And also it's very meaningful, I think, having his daughter, Laura, here. So welcome, and thanks to you and to your entire family for making this possible. I also wanted to congratulate Dr. Lazaroff, and maybe later during Q&A when people ask me questions about the hippocampus, I'll ask you to rise and help me with that. I did a little bit of reading about Sandy because certainly I knew of him, but with age, I've become more fond of taking a historical perspective. And so I saw that he, like my dad, graduated from Illinois, and also really reminded myself of all the fabulous, important science that he did, that he was indeed a pioneer and an innovator. And I think part of the connection, hopefully people don't feel it's too much of a reach, it was clear to me from his career that he recognized the importance ultimately of doing research that you could connect to really changing the way we care for patients, because ultimately all of us in this room today, I think, that's what we're about, is improving lives, lives of the people who suffer from these illnesses. And so the title of my talk is Translational Research in OCD. And, even if you found your way into the audience and don't care anything about OCD, I like to believe that OCD is sort of an interesting model of some of the ways in which we've sought to conduct translational research in psychiatry over the last quarter century or so. So let me just say I don't think I have any meaningful disclosures, though I will be talking about, in fact, mostly about unapproved uses of various treatments, and I'll try to highlight that when it happens. I could spend a whole talk just about translation, what people mean by translation. I try not to get bogged down too much in the terminology. People have thought about it in different ways and broken it up in different ways, but whether you hear the expression from animals to man or from bench to bedside, it really refers to us translating from more basic research all the way to better care. There have been many schemes, T1, T2, T1, T2, T3. One way to think about it is T1 is sort of from basic research to clinical, and then T2 involves real dissemination so that patients are influenced by it. A few more definitions, just because I think it's good to remind people about these terms that sometimes get used interchangeably. That etiology really refers to the cause of a disease, whereas pathobiology or pathophysiology refers to the mechanisms. So the way I like to think about it is that when we learn about etiology, it may give us our best leverage on prevention to stop a disease from being caused. And when we learn about pathobiology, it may give us the best leverage on developing then treatments that relate to that pathophysiology, ultimately aspiring to cure. So today what I'm going to try to do, and I hope it's not too much of a whirlwind, I know well enough that when the calendar says 90 minutes, that at a grand rounds you're probably left with more like 45. So I hope I do okay. As an executive, I should be able to manage time, but we'll see how I do. In whirlwind fashion, I'm going to say just very briefly about the OCD clinical phenomenon. A lot of the talk is going to be about neurocircuitry, as I am an imager. And then I'll briefly touch on one wonderful translational example in psychiatry of D-cycloserine, a little bit about how neurosurgery and neuromodulation seeks to leverage circuitry knowledge. Just a slide or two about optogenetics, where I'll be bringing Coles to Newcastle, where the very best work in this was done here. A line or two about leveraging genetics, and then wrap up. So that is the whirlwind that we have in store. Okay, so I imagine most people here, given the strength in OCD, this is probably not new to you, but OCD is a disorder characterized by obsessions, which are the intrusive, unwanted thoughts, and compulsions, which are the repetitive behaviors typically performed in a ritualized fashion to combat those thoughts and feelings. It's a disorder that's much more common than was thought over a quarter century ago, with prevalence estimates of 1% to 3% worldwide. We do have first-line treatments for OCD that are considered effective. Those include serotonergic reuptake inhibitors and cognitive behavior therapy. But I always like to remind people that for the most part, the rule is residual symptoms, so that 60 to 80% of patients nominally respond, which tells you that 20 to 40% really are not responding, and that of those that respond, they typically have residual illness and suffering. Neurobiological models of OCD have focused on frontostriatothalamic circuitry, whereas models of anxiety disorders have tended to focus on amygdala cortical or amygdala frontal circuitry, and that's what I'll be talking much more about later. I should just flag, and it's only fitting, with Blair having introduced me, since I think we were shoulder to shoulder in the DSM committee, that DSM-5 has seen fit to break out the OC and related disorders into their own category, but still, I think, with people respecting the relationship to other anxiety disorders. So again, this is just a high-level scheme that in general, when one thinks about sort of evolving schemes and strategies for translation in psychiatry, one can think about a neurotransmitter focus where knowledge about pharmacology can lead to classic pharmacotherapies, that a neurocircuitry focus through brain mapping provides information that can be leveraged to advance neurosurgical and neuromodulatory therapies, as well as potentially novel pharmacotherapies. And then finally, what I will talk a little bit about at the end is that with genetics, sometimes we can develop molecular targets that also will enable the development of new therapies. So I'm going to spend a couple of minutes on this slide, this cartoon, which is a schematic of the striatothalamic loop, if you will. And I like to always draw on a little bit of an analogy. Maybe I like this one now because I'm the president of a hospital. But you know, when we talk about frontal cortical function, you hear people talk about executive functions, and so you can think about higher level functions. The striatum in this map is a little bit like a good executive secretary. That is, the striatum carries out automated functions outside of view of the executive's office in a way that makes the executive function much more effectively, and I'll elaborate on that. As you can see, the thalamus is a little bit like the receptionist. We have a famous senior neurologist in the Harvard system named Marty Samuels who says, nobody speaks to the cortex without first speaking to the thalamus. And that's almost true in all cases. But so think about information flowing in through the thalamus to reach the executive's office, and then it gets passed out through the thalamus to action. And that input and output gets modulated by what that executive secretary is able to do. So now I'll hopefully extend but not belabor the analogy. So let me tell you what it's like when my secretary is on vacation, right? Or for others of you, if you have an incompetent secretary, which is what often happens is things make their way into the executive's office that otherwise would be screened out. There are intrusive events that I experience on a regular basis if my secretary's not around. And so that's the metaphor that I would use that maybe is appealing to think about OCD, that if there's something wrong about throughput at the level of the thalamus because of failed governance by the striatum, what you would expect would be intrusions into the executive's office that should have been managed in an automated fashion, and that moreover, there may be other adverse events that have to do with the way in which information flows out of the executive's office to action. So now getting back to the circuitry of it all, there is both a direct pathway here in green, so named because of the direct pathway from the striatum to the globus pallidus interna that has a net augmenting or amplifying effect of throughput at the level of the thalamus, and the so-called indirect pathway here labeled in red, again so named because there's an indirect path from the striatum to the GPI, has a net inhibitory effect and hence has a filtering function. And as the theory has evolved in OCD with these different nodes as implicated in the disorder, the notion is that OCD has something to do with failed filtration and perhaps augmented amplification such that there are these intrusive events and with pseudo-adaptation of ritualized compulsions in an effort to achieve filtration at the level of the thalamus. So let me move from a cartoon to a schematic with actually brain slices, and it also helps orient maybe those in the audience who are not so used to looking at brain slices. So first, let me just ask, these are just transverse sections through MRIs. Just by a show of hands, how many people generally know what they're looking at when they're looking at a transverse section through an MRI? Okay, so it's probably maybe barely half, less than half. So let me just take a second here. Imagine that you are sitting at the foot of my bed and I am laying on my back in a hospital bed. And you imagine an imaginary microtome or guillotine coming down through my head from forehead to back of my head, and then you're peeling away my body and everything, and you're just looking up into my skull cap. That's what you really have here. So, so this would be my forehead. This is the back of my head, right side, left side. This is just at a different level through the brain. This is just over my eyes. This is about midway up in the brain. And this just shows you the different territories, the different nodes of the circuit that are implicated in OCD of frontal cortex, in particular the orbitofrontal cortex, so named because it's over the orbits, over the eyes, through the striatum and specifically the caudate nucleus, then to the thalamus through the pathways I just showed you and back. And again, the notion is that this circuitry is misbehaving in some way, and in particular, it may due to failures at the level of the striatum. This is another set of regions that I do just want to mention. They're coded here in kind of a green color, the so-called paralimbic belt. This is a slide from Mesalem. And I particularly, again, it shows orbitofrontal cortex, but I just want to direct your attention to cingulate cortex, and in particular anterior cingulate cortex, which becomes a very important player in this story as well. One way to think about paralimbic cortex is that paralimbic cortex, as could be said for the whole limbic system, sets priority for us. So in some ways, the functions of all the different subterritories of paralimbic cortex tend to set priority. So, you know, I've had the good fortune, you've heard me mention my dad a couple of times. I often like to say to an audience, so, you know, my dad was the first geneticist at UMass. He passed away a few years ago, was a lifelong, grew up in New York. But what I like to say about him is he entered the field of genetics before the double helix, and he lived to see the completion of the Human Genome Project. So just think about that for a second. So, you know, all of us who devote our lives to science, you know, you pray for sort of the good fortune to live through and hopefully contribute in some way to a kind of movement that really changes the world. And I feel exquisitely fortunate to have been able to be drawn through fellowship work and then much of my career trajectory up until a few years ago to the field of neuroimaging. In the same way that Sandy was clearly interested in the interface of cardiology and psychiatry, then in cardiology we saw, you know, going from probably raw auscultation to, you know, the stethoscope to things like an EKG, an electrocardiogram. These neuroimaging tools in psychiatry really have given us the capacity to non-invasively, safely measure indices of chemistry, structure, and function. And really for the last 25 years, a lot of the advances, at least to the level of meaningful heuristic for our field, really have been born out of psychiatric neuroimaging, among others. What's kind of cool about the story of OCD is virtually all of these different methods have been leveraged, and virtually all of them have yielded broadly convergent evidence about this same circuitry being where the action is. So there are morphometric MRI methods, that is methods where we look at sort of the size and shape of different brain structures. There's DTI, which allows us to look at some of the coherence of the white matter tracks, the wires that connect the nodes in the circuit. Spectroscopy is used to measure neurochemistry, but there are neurochemical indicators of neuronal health that I'll tell you about. And then functional neuroimaging that gives us indices of gross brain function has been employed in conjunction with various types of experimental paradigms so that we can study people nominally at rest to see what their brain is doing. We can look at changes in brain activity from pre to post-treatment, with the notion being that if there's an abnormality and it moves towards normal when successful treatment happens, that implicates the brain region. Symptom provocation, where graphically we can see how brain areas become more active in league with symptoms, suggesting that they're mediating the symptoms. And finally, cognitive activation, which is a little bit like a cardiac stress test method of probing certain circuitry under certain conditions. And so really all of this has been pertinent to the advances in OCD. So in Whirlwind, I'll just say the morphometric MRI studies of OCD have probably been least consistent. By and large, findings in striatum, like the caudate, predominate, but especially in large-scale studies, we've seen other regions affected, like orbitofrontal cortex, the thalamus, and the amygdala, and even the hippocampus in some meta-analyses. So the structural studies fit with the model, but I don't think they've been the most compelling element. There are convergent evidence to suggest that, convergent data to suggest that there are white matter abnormalities in OCD. This is probably the area, given that DTI is a relatively new tool, that is also sort of less mature, if you will. From very early on, there were indicators of abnormal total white matter volume. Also, the various tracks that connect the node of the OCD circuit have all been implicated. And perhaps most germane to a later part of the talk is that it's these very same white matter tracks that have been targeted in ablative neurosurgical procedures that I'll be telling you more about. And I will come back to this at the end when I note that some of the genes that regulate myelination have been implicated in OCD. So again, the very earliest studies using MRS, and with MRS, you select a volume, you generate a spectral pattern that allows you to calculate by area under the curve the concentration of certain chemicals in the brain. So N-acetyl aspartate, which is an indicator of healthy neuronal density. There have been findings of abnormalities, again, in nodes of the OCD circuit. One of the principal neurotransmitters of the OCD circuit is glutamate. And so glutamate is another MRS visible compound. And so abnormalities, actually elevations in glutamate at the level of the striatum, have been found that correlate with symptom severity and return towards normal following successful treatment. So again, this in particular is a glutamatergic projection, as are some of these, and these as well. Okay, so as I indicated, the implicated regions are orbitofrontal cortex, anterior cingulate cortex, and the caudate. We've seen by the neutral state resting studies that those with OCD show higher activity in all the nodes of this circuit at rest or at baseline. From pre to post treatment, what we see is that with successful treatment, that hyperactivity is attenuated, and that then with symptom provocation, if you take individuals with OCD at rest and image them, and then image them moments later after provoking their obsessions and compulsions, you see accentuated hyperactivity in this circuit. So here an example of just some of the historic studies in this regard. It's the UCLA group with Lou Baxter and his colleagues that were probably most influential early on to show these this hyperactivity in the caudate, head of the caudate in this particular study that returned towards normal after successful treatment, regardless of whether or not that treatment was a CBT or was serotonergic reuptake inhibitors. So these are just sample images, but again, in a statistically significant way, those changes were found. This is our own work back in the early 1990s when we began using the approach of symptom provocation. And I have to admit, seeing these statistical images come out of the PET scanner were one of those eureka moments. This is a slide that's followed me around for whatever it is now, 25 years, 23 years. So graphically, this was a group of 8 subjects who were exposed right in the scanner to contaminants and other things that provoked their OCD symptoms. And the differential activation was in orbitofrontal cortex, caudate nucleus, the thalamus, and the anterior cingulate cortex, all the elements just lighting up like a Christmas tree. And you can see how relatively silent the other areas are in terms of differential activity, and certainly so in a statistically significant way. Over the years, we and others have done, and this is by no means a comprehensive list, though as of 1996, close to a comprehensive list of symptom provocation studies in OCD and other anxiety disorders, as well as inducing anxiety in healthy controls. And what I just want to highlight is these paralimbic areas that tend to much more non-specifically activate when you make people anxious or sympathetically aroused. I know, Joanna, we had that discussion with Tim in the morning, that paralimbic areas you tend to see pretty non-specifically come on board. But it's interesting that the anterolateral orbitofrontal cortex and caudate seemed much more specific and really consistent across OCD studies. Again, kind of underscoring the notion that maybe there's really something specific about OCD that relates to these elements of the circuit. This is a great meta-analysis, actually, and comes from a couple of chapters, really by Roll's group, though it's, I guess, Kringleback and Roll's. It really gets to us across many, many imaging and other types of clinical neuroscience studies. It turns out that orbitofrontal cortex is not monolithic, and that medial orbitofrontal cortex tends to be more positively valenced material, whereas anterolateral orbitofrontal cortex mediates more aversive and negatively valenced material. So again, just to remind you, OCD involves activation of these more anterior and lateral orbitofrontal areas. So again, think of sort of the negatively affectively tinged aspects of obsessions. So hopefully I've given you sort of by brief and whirlwind backdrop some of the reasons why we believe that corticostriatal circuitry is implicated in OCD. One of the questions is what aspect of corticostriatal function is really especially pertinent to OCD. And in our lab, we pursued the notion that it really had to do with this automated non-conscious processing aligned with the story I told you about the incompetent secretary. And so really many years ago, and it was in the context of my K Award, right? So I know I spoke with Joanna and others about the K Award trajectory, this notion of trying to develop a task for the scanner that would allow us, like a cardiac stress test, to really activate corticostriatal function, but in a specific context, in the context of more automated information processing. And to tell this story, I need to digress just for a moment to acknowledge that there are many different types of learning and memory. And here you can see that there are both explicit, that is conscious forms of learning and memory, episodic, that is remembering events, semantic is remembering facts that we're consciously aware of. Then there are implicit, that is non-conscious ways in which we learn and our behavior changes. One of them is by skill learning. So if you think about it like the ways that we learn to ride a bicycle, there are elements of that that are really going on non-consciously. And it turns out there's really quite good evidence that this implicit skill learning is mediated by corticostriatal systems. And just to give you some indication of this, for people with primary basal ganglia disorders like Huntington's disease and even Parkinson's disease, very early on in their disease, their explicit learning is intact, whereas their implicit learning is impaired as some of the first deficits. Whereas in contrast, classical temporal dementias like Alzheimer's disease, where the medial temporal lobes are affected, very early on, implicit learning is intact. In fact, at MIT, I had the opportunity to meet the famous patient HM, who by virtue of ablation of the hippocampus, each time he would come to the lab, wouldn't recognize any of the people who he's been meeting with for 30 and 40 years, but actually could get better at skills each time he trained on them because he had intact corticostriatal circuitry. So just to remind you, there's fabulous work done in the mid-90s and since using cognitive neuroscience and functional imaging. This is out of the work, this is from the lab of Dan Schachter at Harvard, showing really in a very exquisite way how the hippocampus mediates this, remembering word pairs or remembering various facts. And so now I'm just going to tell you about the task that we utilize to probe corticostriatal function. This is called the serial reaction time task, and to the subject, it looks like a simple continuous performance task. So imagine that I use these 4 fingers to correspond to 4 individual buttons while I'm sitting in the scanner, and a cue comes up like this in one of the 4 positions, and all I do is push the button that relates spatially to the cues. So very simple task, virtual, 100% correct rate for subjects, but unbeknownst to them, we slip in now and then a 12-position repeating sequence. They're not consciously aware that the sequence is present, but they learn it, and they learn it in an automatic way. And so these are just some data from our lab showing that you begin by first doing random order, and people habituate, they get used to the task, and they level off their performance. Then for the implicit trials, what you do is you introduce the repeating sequence. And what you see is their reaction time improves because in an automated way, if you will, their striatum is learning the sequence of movements. Then when you reintroduce random, the reaction time jumps up again. And that delta is the quantitative measure of the learning because it shows that without the sequence information. And then what's interesting is you debrief them, you find out, you confirm they had no idea that a sequence was present, and then you teach it to them explicitly. And when you teach it to them explicitly, they now employ other brain regions, and they really accelerate their learning to a very different asymptote than what's achieved in automated fashion. People get that? What we showed over about a decade was first that SRT studies in healthy subjects really reliably show this striatal activation, but that when we have people with OCD do it, they don't activate the striatum. even though their performance is normal. And what was really interesting and unanticipated is that they activate hippocampus instead, and presumably in a compensatory fashion, since their performance by reaction time learning. Other labs have used other implicit learning tasks and shown the same thing, that is normal performance, failure for striatal recruitment, and aberrant hippocampal recruitment that appears to be compensatory. And we went on to study a bunch of other disorders, and without going into the full detail, what's interesting is no disorders other than OCD have we found to show this same profile: normal performance, failed striatal activation, aberrant hippocampal activation. So, and this is just an example of one slide of data from one of these studies, again, showing the striatal activation in normals, the absence of striatal activation in the OCD group, and this very robust aberrant bilateral hippocampal activation instead. And this is just one of our replications, just showing from another perspective this aberrant activity in the hippocampus. So Dr. Lazaroff, I'll look closely at this slide. I'm going to call on you later to tell me. Now I know they're very meaningful subterritories of the hippocampus, so I don't know if you can tell me from this slide which is. So what is the hippocampus? I can tell you, I found out the hard way. It's actually a very good bar in Paris called the Hippocampus. But just to remind you, it's sort of an interesting thing, and I'm sorry I keep mentioning Dr. Lazarov's name, but I think it's really, really at Columbia as much as anywhere under Renee Henn and others, that there's a much more sophisticated appreciation of the meaningful subterritories of the hippocampus, much more so than when we were doing this work, a decade ago. And I think a fair extension of this work would be to think deeply about what subterritories of hippocampus are engaged. I don't know the answer to that. But at least when we were doing this, the way we were thinking about it is it seemed like the hippocampus was playing a really critical role in managing information within the conscious domain. And isn't it sort of heuristically appealing, given what we know clinically about OCD, to think that, true to the neuroanatomy, there's a failure in the automated information processing, and that instead people with OCD are using machinery that typically renders the information being processed accessible to the conscious domain, so that people with OCD are having these intrusions of thoughts that maybe the rest of us are able to put to rest outside of conscious awareness. So the conclusions from this first phase, and I can see I'm going to be a little bit time challenged, are that anterolateral OFC activity is associated with negatively valenced thoughts and controls and with obsessions and OCD. That reverberation in this OFC, orbitofrontal cortical striatal thalamocortical loop, may underlie obsessions and compulsions, be due to deficits in the striatum, and be mediated by glutamatergic transmission, and that corticostriatal dysfunction may underlie implicit processing deficits with aberrant medial temporal activity paralleling intrusions of thought into consciousness. So it's sort of a set of hypotheses that try to reconcile the data as they have emerged. Now I'm going to switch gears just to talk about amygdala frontal circuitry in OCD, and then really move to the part of the talk where we think about how we're leveraging this knowledge to hopefully develop new types of treatments. So amygdala frontal circuitry, in many ways, functions of the amygdala are very well preserved across phylogeny. And so I think fabulous advances in translational neuroscience have been made precisely because when it comes to fear conditioning and extinction, a lot of the same processes that we see in rodents, we can extend right up through primates to man. So again, this is a circuitry that's implicated in fear conditioning and extinction across anxiety disorders. There's evidence of amygdala frontal abnormality in OCD, some of it emerging relatively recently. And that again, exposure and response prevention therapy in a way leverages, really involves extinction processes. And so this is a very meaningful sort of framework for thinking about OCD, because really one of our best therapies is an extinction-based therapy. So just by a show of hands, how many people pretty much know about fear conditioning and extinction? Yeah, so I did better with that than the MRI. I think all the hands went up. But so just to remind you, you can take an animal, a rodent, expose them to a tone that is paired so as to predict an aversive event like shock. Subsequently, upon learning that association, the animal will respond to the tone in a way that's similar to the shock in preparation for the shock. And you can see that, so freezing is the measure of sort of anxiety, if you will, in the case of the rodent. So you can condition them, fear condition them to respond to the tone alone. Then if you present the tone alone repeatedly with no shock, they extinguish. That's an overwriting. It's not an unlearning. It's a learning of a qualification that now in this new context or in this new time, tone no longer predicts shock. And part of how you know that is that if you return them to the unsafe context or away from the safe context, you get full return of the conditioned association. But what's cool is in the safe context, where you now have repeatedly exposed them to tone with no shock and extinguished the result, if you bring them back a day later, they retain that extinction knowledge. And again, it's context gated. What's so important about that is that the machinery in the brain to do this is a little bit different than the machinery of the brain to do this. And if we want to be healthy in the world, when we extinguish to a given stimulus, we really want to make sure that when we come back the next day, we are still extinguished to that stimulus. And so again, for those of you who engage in cognitive and behavior therapy, you get that really one of the big deals is, you can do exposure and response prevention therapy, but at the end of the day, its efficacy is really going to be grounded in whether the individual patient is able to retain and generalize that extinction information over time. So again, the neurocircuitry of that paradigm is exquisitely worked out thanks to people like Joe Ledoux and Mike Davis and others. And here I'm just going to give you the short course that the amygdala plays a very critical role in the formation of the association, that is fear conditioning, as well as the acute process of extinction. But there is a territory, thanks to Greg Quirk and others, called infralimbic cortex, which plays the critical role in extinction recall. That is, retaining and being able to recall that extinction information days into the future. So again, amygdala plays a critical role in conditioning and extinction, infralimbic cortex in extinction recall. And so what's kind of cool is we went and Mo Malad, who was a graduate student in Greg Quirk's lab, who did really the pioneering work to map out IL, infralimbic cortex is the key area for extinction recall. He left Greg's lab after he finished his postdoc and came to us and modified the paradigm for humans so that we could actually do fear conditioning extinction and extinction recall in the scanner. And we, pretty much contemporaneously with Liz Phelps and her group, beautiful convergence across a series of studies that the human homologue of IL, that is that which mediates extinction recall, is this ventromedial prefrontal area that's contiguous with medial orbitofrontal cortex. So remember the roll slide I showed you? Medial frontal cortex is sort of good. It mediates extinction. Anterolateral orbitofrontal cortex is bad. That mediates the more aversive thoughts. And so this then, the notion is, this is a homologous region in humans that we should think about as a potential target for modulation. So again, mapping out that the human homologue, being able to translate from the rodent studies to humans, that this would be the equivalent target, if you will. The evidence in terms of sort of amygdalocortical abnormality in OCD, it turns out that for all these disorders, if you expose people to sort of provocation, you'll see increased activation of the amygdala, not surprising. In general, when people are afraid, you see amygdala activation. It's interesting that when you show people general threat, that the different disorders have quite different profiles, which I won't say more about other than that that's sort of interesting. This is from sort of a meta-analysis that Mo and I did together, just sort of demonstrating, so this very same postural medial orbitofrontal area that we had shown was the human homologue for extinction recall, how imaging studies using other paradigms have consistently showed sort of low or deficient activity in that region across the whole range of different anxiety disorders. each of which is generally potentially treatable by extinction-based therapies, but that fundamentally these disorders may suffer for some deficiency in this region that mediates extinction recall. Well, actually, I'm going to go back. But to also highlight what was unique to OCD is the robust way in which anterolateral orbitofrontal cortex is hyperactive, something we're really not seeing in any of the other anxiety disorders. There have been a few studies done now looking at extinction recall in OCD. This is the most recent one, I think, in JAMA Psych 2013, again, from Mo Malad's lab, again, showing this very pronounced failure of extinction recall in people with OCD. Again, just demonstrating that is a real sort of physiologic attribute and behavioral attribute for people with the disorder. So we'll take it one step further. Again, this comes from Greg Quirk's lab. So I told you, again, IL is, I hope you don't mind me saying it this way, IL is good. It mediates extinction recall. They found an adjacent area called PL, prelimbic cortex, that does just the opposite. It actually exaggerates, it augments fear expression. And it turns out that these two regions in Greg's lab, he was able to show that they're mutually inhibitory. So that the more you have stuff going on that activates PL, you augment fear responding. Whereas the more you can employ IL, that gives you what you need to suppress that, and particularly in the context of recalling extinction information. So what's cool is we had human data sets that allowed us to look at whether there was a human homologue of PL. So both we had structural data from the very same healthy subjects who had been engaged in various fear conditioning extinction paradigms, and we showed this very powerful correlation of cortical thickness to fear expression in this dorsal anterior cingulate area. And then by functional MRI from our conditioning and extinction recall, we found actually in different cohorts of subjects the very same sort of territory. So fear expression correlated with dorsal ACC thickness and activation in healthy human subjects, suggesting that this might be the human homologue of PL. That is the bad area, okay? So for amygdala-frontal circuitry in OCD, amygdala-frontal circuitry is implicated in fear conditioning extinction. IL in rodents, that is medial orbitofrontal cortex in humans, mediates extinction recall, appears to be deficient in some way in OCD. PL in rodents, homologous with dorsal ACC in humans, may increase fear expression and oppose extinction. There's evidence of amygdala-frontal abnormalities in OCD, human OFC insufficiency associated with deficits in extinction recall, dorsal ACC hyperresponse associated with increased fear expression, and there's a whole nother literature on false alarms in the dorsal ACC I'm just not going to go into today, principally from the group in Michigan, of, again, findings in that same territory in dorsal ACC that is really the human homologue of PL. And extinction-based exposure response prevention therapy relies on this very same circuitry. So I'm going to pause just for a second. I know I probably have 10, 15 minutes max left, something like that, 15. So I'm going to pause there because I hope what you see, though it's been whirlwind, is a very deliberate way of using different approaches that are between basic and translational to really try to understand neural underpinnings of the disease, as well as, to some degree, human functions that we think are pertinent to the disease. And I hope you can see, because that's what I'm going to build on in this next part of the talk, how having that knowledge gives us translational leverage to think about posing hypotheses and trying things about what might utilize that knowledge to advance different treatments for these disorders. So one of the very cool ideas that I think in fairness came out of Mike Davis's lab, and I think I was almost like standing there when like the light bulb went on over his head. It was at an ADAA. It was one of the special research things where we brought basic science people to hear about exposure-based therapies. This notion that if you had a drug that would augment extinction processes, you could give that drug to somebody who's doing exposure response prevention, and you could accelerate or fortify the process. And that's really just the idea that Mike pursued. And in fairness, with Kerry Ressler as the lead author and really the landmark study, that is, it was really, I think, already known to Mike that D-cycloserine, a very old drug, a glutamate agonist that binds at a glycine site, it was used to treat TB, but very well tolerated, had the profile that should make it facilitate extinction processes. And indeed, it was shown 2002 that DCS facilitates extinction of conditioned fear in rodents, and that it does so even if it's given systemically, which is of course important if you're thinking about a treatment. And they then proposed the potential use for augmenting extinction-based behavior therapy. And Kerry Ressler did the initial study. I'll show a slide from that in a minute. And then following that, others followed suit in other anxiety disorders where extinction is used. So these are data from the very first study that Kerry led along with Barbara Rothbaum, Mike Davis, and others at Emory, where they trained people on a virtual reality. They had people do a virtual reality-based exposure task for people who were height phobics. They had a purposely subtherapeutic clinical intervention, and then gave some people that subtherapeutic exposure response prevention, really exposure therapy, with and without DCS, and showed, again, accelerated response with DCS. We in our own lab, together with Sabina Wilhelm, who's known by some of you, tried the same thing in OCD. So what I would say is that a lot of the examples I'm going to give you are really not home runs. So DCS was a really cool idea, and now, over a decade later, people have played around a lot with it. Dose finding, what's the optimal dose? What's the optimal timing? You could actually give it right before the CBT. There were ideas of giving it after the CBT, after you made sure that the session you had just given was an optimal session. And people have tried it in different age groups. I think the punchline on this is it's clearly not a home run. A lot of the treatments, as they've really moved to controlled trials, have been sort of underwhelming. And I think there was a pretty definitive study this past year in children that while there was eagerness or sort of optimism that this would be useful in youths, Storch and colleagues conducted a pretty large-scale, well-controlled study that yielded a negative finding. So here, this is my time to emphasize DCS is not approved therapy for OCD and anxiety disorders. This is not one that I would sort of take to the bank, but it's a great translational story. And it may still be that there will be a subset of patients or disorders for which this is meaningful. We still use it in our OCD Institute now and then, for people who are otherwise treatment refractory. The data seemed promising that it would accelerate response, and in animals what's so cool is it fortifies the response. It actually makes the extinction less prone to reinstatement. So I think in some ways, as I put in the early schematic slide, if you have circuitry knowledge, it seems like the best way to leverage that knowledge is something that gets in there and changes the activity in the circuitry. So neuromodulatory and neurosurgical therapies in some ways seem like the most rational and natural sort of rejoinder to the information that we have that is circuitry based. So psychiatric neurotherapeutics is one term that's used for these various types of treatments. I think people in this audience have certainly heard of ECT, vagus nerve stimulation, transcranial magnetic stimulation, but I'm mostly going to focus on psychiatric neurosurgery, and in particular both the historical ablative procedures, because they're so graphic in terms of how they have sort of targeted different brain regions and circuits. And then most recently, and what I think there's still a fair amount of excitement about, is deep brain stimulation, which is a much more targeted and adjustable, reversible methodology. Again, here you see in red at the bottom, neurotherapeutics are not approved for OCD. I believe there is still a humanitarian device exemption in place for DBS for OCD, but these are not technically, I believe, approved uses. So there's a long history dating back now probably a good 30 years or more for ablative, targeted, stereotactically mediated ablative procedures in psychiatry. I can't emphasize enough, we're not talking about sort of gross lobotomies. We're talking about targeted psychiatric neurosurgery. And here as an aside, I'll say that I sort of had the privilege of during my residency, I got assigned to the psychiatric neurosurgery team at Mass General Hospital under Tom Ballantyne, who was really the person to pioneer cingulotomies. And for a period of time, I think we were essentially the only site in the United States that was doing these procedures, and it very much became a platform and a springboard for bringing capsulotomies from Europe to New England and then ultimately the United States. And those really were the forerunners of DBS. So here, I don't want to give this too much short shrift, but I think what I will just say superficially is all the targets of these various psychiatric neurosurgical procedures, the targeted ones for which there was evidence of benefit in OCD, are all lesions that are made either in one of the nodes of the OCD circuit or in the connective fibers that connect the nodes of the OCD circuit. And that's true for anterior capsulotomy, anterior cingulotomy, and subcaudate tractotomy, limbic leucotomy, which is actually a combination of these lesions. I'm mostly going to talk about anterior cingulotomy because that's the procedure that I know best and that I was very involved in these sorts of procedures. But I'm also using that as an example because later when we come back, you'll see that there's something about the locus of that lesion that is particularly intriguing, and we have some imaging data on that. So maybe I'm going to go to it right now. So I already showed you these slides, right? Remember, PL, bad, it augments fear expression. So, you know, think about a treatment for the most ill of the most ill, people with OCD, people who are mostly housebound, have been tried on all evidence-based treatments, are typically on the verge of suicide, looking for any alternative to ease their pain. Cingulotomy in that context was a procedure that was developed both for treatment-resistant depression and treatment-resistant OCD that involved these lesions in the anterior cingulate cortex. And so this information was not known and could never have been known at the time this procedure was created. But isn't it interesting that those lesions are essentially at exactly the locus that we now, decades later, have demonstrated is the human homologue of PL? Whoops. So the rodent model suggests that lesioning PL, that is that dorsal area, directly releases IL from inhibition. So it's not just that you're reducing fear expression directly by lesioning PL, but maybe you're liberating IL to be more effective in extinction recall. So if that's true in humans, lesions of the dorsal anterior cingulate cortex, like cingulotomy, could enhance MOFC, medial orbitofrontal cortical function, and actually enhance extinction capacity. So not just reduce the aberrant excessive fear expression, but in some ways liberate and facilitate extinction processes. And this is just a circuitry map from Greg Quirk's work showing this phenomenon where the PL and the IL have this relationship where limiting PL function actually liberates IL function in the service of extinction. And so in fact, in data from the very subjects that we do cingulotomy on, shows even though the lesion is being made in a very distant location from medial orbitofrontal cortex, that in our patients who receive cingulotomy, they show increased activity in medial orbitofrontal cortex. So again, the idea being that cingulotomy may confer its therapeutic effects by lesioning the human homologue of prelimbic cortex and liberating, thereby enhancing the homologue of infralimbic cortex. So sort of fast forward to deep brain stimulation, which is of course a very exciting technique because though still clearly invasive, it's the ability to adjust and reverse, unlike an ablative procedure, makes it a much more appealing way to intervene. And it was really Bart Nutin and his group in Belgium that pioneered this for psychiatric uses, and in particular for a very small series of OCD cases to start with beneficial effects. Sorry if this doesn't quite show up well, but this area of the internal capsule Some of the ventral contacts involve some of the very same sort of corticostriatal pathways that are being initiated from orbitofrontal and anterior cingulate cortex. And here are just some clinical data. I would refer you to the papers by Ben Greenberg and Damien Denis and others that have really been forging forward. The Mass General currently has, I think, about a $40 million grant from DARPA to be moving from this conventional type of DBS to what's called closed-loop DBS. The notion that you would have these implantations, but the implantations would actually be recording from cells so that the stimulation that is being provided is actually governed by the nature of inherent activity in the brain. I think it's immediately apparent to people how that could be very helpful for treatments in the context of seizure disorders, where, being prompted by some type of activity in the brain, you trigger, not unlike, I guess, implantable defibrillators, the same sort of idea for brain. So, the science fiction of yesterday becomes the reality of today. So I would just say, being involved in some of the deep brain stimulation for OCD, there too we saw that with acute stimulation, even though the site of stimulation was distant from medial orbitofrontal cortex, again, accentuated activity in medial OFC. And it's so interesting. In the case of OCD, unlike depression, DBS really didn't render instant relief of symptoms. The notion was that it enhanced the trajectory towards relief of symptoms over days to weeks. And so I know that Ben Greenberg, among others, really believed that there was some way in which it was rendering these patients previously intractably unresponsive to CBT, now able to respond to other treatments. So why would enhanced medial OFC function ameliorate OCD? By counteracting the deficits in medial orbitofrontal cortex, by facilitating extinction recall capacity, which in turn might facilitate effectiveness of exposure response prevention. So in conclusion, in terms of neurotherapeutics and OCD, surgical and device-based treatments hold promise as therapeutic options for severe, otherwise refractory psychiatric disorders such as OCD. This treatment approach is well suited to leverage advances in translational neuroscience and neuroimaging. In particular, emerging data suggests that medial orbitofrontal cortex and dorsal ACC may represent fruitful targets for neuromodulation with respect to OCD and other disorders. Anything that stimulates or augments MOFC function could enhance extinction, and suppressing DACC function may mitigate condition fear acquisition and expression. So I am actually in deference going to give optogenetics short shrift because this is the place where you guys have done the coolest work. I was privileged to be invited to write a little commentary when Blair and her team, Dr. Amari and colleagues, published a fabulous original report for appearance in Science, contemporaneous with sort of a complementary report out of the group at MIT. I think Dr. Bergeri, though I hesitate to try to pronounce his French name. By a show of hands, how many people know about optogenetics? Oh gosh, all the hands need to go up. I'm gonna take a second on this. So Carl Diceroth and others developed this unbelievable space-age technology, which basically, leverages basic biology like from algae, where there are entities that are light responsive and differentially responsive to different wavelengths of light, such that it is possible through essentially transfection to introduce into specific types of cells the capability of being selective wavelength of light responsive. Once you do that, so you can introduce to any node in a circuit that phenomenon, then by piping in light, targeted light, you can turn cells on and off. And so this technology was used to modulate corticostriatal circuitry, either to induce through repetitive stimulation of corticostriatal circuitry a picture that looked like OCD, or to actually utilize this very targeted neurostimulation to relieve OCD-like symptoms in a genetically modified SAPAP3 strain of rodent. And so now I apologize for giving this short shrift, because as I'm looking at the faces, I'm getting that people actually don't know this science. So let me suggest you guys should do a grand rounds on optogenetics and your own work, because I actually think it's the most exciting stuff, but I'm giving it short shrift because I'm not the expert in the room on this one. So I think I'm going to more or less finish up just by a tip of the cap to genetics. Obviously, you know, some of the great advances and potential advances in our field will come from genetics as we understand genetic anomalies that underlie our disorders. And here I just want to mention, to the degree that there have now been a number of really massive genetic efforts in OCD, I think the best brief summary of this literature is that there are a subset of genes that have been implicated that are associated with glutamatergic function. There are a set of genes that have been implicated that relate to myelination regulation, and a sort of a family of genes that, that have to do with regulating synaptogenesis. But I'm just going to zero in on this one, SAPAP3, which again, as I said, is the very same one that Guopeng Feng and his colleagues at MIT in that tandem study to the Amari Columbia study used. What's cool about SAPAP3, let me see if I'm going to get to this in the next slide. Yeah, SAPAP3 knockout mice very reliably, with 100% penetrance when they pass adolescence, develop a compulsive behavior where they sort of chew off and scrape off their own fur. So it's believed to be, you know, a reasonable analog. I should just say the bulk of large-scale studies, actual clinical studies in humans, don't seem to implicate SAPAP3 as a common variant to underlie OCD as a disorder. But it's a very interesting model because it's known to be a striatal glutamatergic scaffolding protein. And so for us, just see what I have here on the slide. So repetitive grooming behavior ultimately resulting in severe skin lesions. Yeah. So what Guopeng, who is the guy, Guopeng Feng at MIT, who actually characterized these animals, is that they have reliable striatal hyperactivity like OCD. Their behavior is actually responsive to fluoxetine, which is a first-line, you know, pharmacotherapy for OCD. And again, this was the same group that then used the corticostriatal optogenetics to sort of cure, if you will, this. So in collaborating with Guopeng, what we've done now is, you know, we have a 9.4T MRI device that allows us to do both spectroscopy and structural imaging in rodents that have been genetically modified. And so for us, what we think is so exciting about SAPAP3 is you have animals where you're going to have 100% penetrance. And so what you can do is you can begin to study their brains non-invasively, serially over time before they manifest the symptoms. And this is what I really meant about, you know, trying to leverage something about biomarkers and etiology to think about prevention, or at least early intervention. So we've now published sort of a brief initial series of studies, and we have funding to extend this work, in essence showing that there are specific MRS abnormalities visible in these animals before they begin to show their symptoms. So we think that's a really exciting thing to think about, you know, for use who might be at familial high risk, to think about methods of imaging them to identify who will develop symptoms in advance of symptom development with the potential for early intervention. So in conclusion, I hope I have outlined for you and that you believe that translational research in animals and man have implicated corticostriatal and amygdalofrontal circuits in OCD, and I give you a review article if you want to read more about it. Circuitry knowledge may be leveraged to develop neuromodulatory treatments, and I think that is going to be a very exciting direction for the field of psychiatry in general and OCD in particular. Refined animal models also provide vehicles for advancing behavioral and pharmacologic treatments for OCD, and I think the DCS story is a compelling one, even if DCS itself is not a home run. Advances in genetics may provide opportunities to develop prevention or early intervention strategies by identifying individuals at risk. Moreover, genetic knowledge may provide clues to new molecular targets for diagnosis and therapeutics. And I have the sense, I hope it's not an overreach for me since I did not know Sandy well. I have a feeling that if Sandy was alive now or was a youngster now, and the trajectory of his career was forward from this point, this notion of having genetic knowledge that could steer us to new molecular targets for clinical trials is something that he would probably be very excited about. So I thank you all for your attention. I think I've left a few minutes for questions, if there are any. So again, thank you.
So great. Wait, is this working? Thank you. So thank you very much, Scott. Thank you very much, Scott, for that incredible tour de force. So we get lots of lectures on biology and lots of lectures on imaging, but it's not so typical for someone to bring it all the way from the imaging all the way to the clinical therapeutics. So the floor is open for questions. We have a few minutes. Go ahead, Alex.
Thanks. I'm trying to sort of keep everything straight. You spoke about deficits and extinction in OCD patients,
Yes.
and you spoke about focusing on extinction for therapy, and this homologs for IL and PL and their dysfunction. Now, with the capsulotomies and the deep brain stimulation, you have normalization or improvement of function in these two areas. What happens to the extinction in these patients?
Yeah, so that is a great question, and I actually don't know whether as part of the DARPA grant whether they're doing that. I know Mo Malad's at Mass General at the very place. I also know that they were doing gamma knife capsulotomies. That was the pioneering piece that really at Rhode Island Hospital that Ben Greenberg and Steve Rasmussen and that group leading the way. And I think in tandem, similar work was being done in São Paulo, Brazil. Both of those places, we collaborate with all those guys. They got that the conditioning extinction recall was a really interesting paradigm that that Mo developed. And so I think to varying degrees they have begun to look at that. But I actually haven't seen anything published yet that specifically speaks to that issue. I mean, I think everybody here can probably appreciate, it's hard if you're talking about very severely ill individuals who are typically coming from distance to get what they think is going to be a life-saving procedure, and that's a surgical procedure, that all the things we do with them, for them, to them, just as part of the core treatment represents really a pretty heroic subject, I'll say patient burden. And so which things you choose to do. I mean, what I didn't go into depth in was really just the challenges associated with each of these paradigms. So there are all kinds of challenges around the serial reaction time paradigm to make sure that people don't learn the sequence explicitly. With the conditioning extinction recall, I mean, that is a 2-day psychophysiology paradigm. It's, when you're getting healthy college students from down the road, it's a walk in the park. When you're talking about people who are really profoundly ill, it's really hard work to do. So it doesn't surprise me, even though it would be a natural, rational, logical, important thing to do, that nobody's done it and published on it yet. So sorry if that was long-winded, but and I do think that's exactly the right question to ask. I should have also said.
Other questions? All quiet.
You let me off easy. Is Dr. Lazaroff still here?
So let me ask a question around, so Josh Gordon was here last week, and he was talking about sort of his vision at NIMH and towards, circuit-based psychiatrists. To some extent, you gave us a talk. I mean, he ended in a sort of futuristic mode of maybe one day we'll be doing viral vectors in people and turning on and off targeted circuits in humans, although acknowledge that to inject anything into a human hippocampus is a major issue as well. But you actually gave us something that's not so far in the future. It's already being done. But I'm sort of curious if you think forward in terms of, so if you think forward, what would you predict?
Yeah. So, I mean, I think if I start big pictures, I think you're inviting me to, you give me an opportunity just to make a couple of points. One is I think anytime you're trying to do pioneering work in therapeutics, risk is one of the huge dimensions, right? So the more you have something that might make a huge difference and it's safe, you can run pretty far, pretty fast with that. The more invasive a procedure is, so it's really been the problem with neurosurgery, is how do you move neurosurgery forward? Because, by definition, you're talking about non-trivial procedures, even with something like DBS. And so part of why I lead with that is, we would love it to be the case that you could get these kinds of good effects with something like TMS that's, pretty clearly safe, less invasive. I guess the other important point to make is that with all of the neurostimulation treatments, you have almost infinite parameter space, right? So, we come from a background of, and maybe this is not fair with CBT or certainly pharmacotherapy, we're kind of talking about dose and frequency. It's a pretty tight, not oversimplifying, but mostly it's a pretty tight space to explore, dose finding. For neurostimulation, it's nothing like that. You've got about 5 or 6 parameters. They're continuous functions. You have no clue where to start. And then if you add to that it's risky, that makes it really hard. So with TMS, there were some early open trials that sort of hinted at TMS maybe having some fruitful targets in OCD. I don't really believe, my current reading of the literature is I don't really believe that any of that is yet panned out in a very compelling way, but that's far less invasive. And so I think, that's really the issue is, whether you're injecting viruses into the hippocampus or you're, implanting electrodes and power packs into people's chests and skull cavities, that it's hard to believe that that's ever going to be a first-line treatment. God help us if that's ever a first-line treatment for anybody, right? So now, on the other hand, you don't have to sit with many individuals or families who are dealing with, life-constraining, profound, severe treatment refractory illness to say it's worth doing exploring those heroic options. But as we talked a little bit about, I think in both of my groups over the last 2 days, I'm now like a hospital administrator. So I'm thinking about, what's gonna better treat populations. So, I think we need to, probably invest at every level, the heroic things, the things that will get the treatments we already know work to the people who need it, because we're not so good at that yet as a nation, to the things in the middle. But I think having potential new treatments where we have some confidence that they are of low risk, it really gives you a very different opportunity.
Blair?
Yes.
Thank you, Scott. That was lovely. But to keep you on the heroic treatments, about which I'm a rank amateur, could you comment on whether there are differences between DBS, for example, or such invasive therapeutics, for OCD versus refractory depression? Is there specificity or not?
So I think you'll correct me, Blair, I think the number we throw around is that people with OCD, about 2/3 of them have depression. I can tell you that if you go to the most treatment refractory group, it's probably closer to 100%. And so I think part of the confound in this is at least in that direction. When people have tried heroic treatments for OCD and patients get better, the truth is if it's a heroic treatment for depression, they may well see that, even if it's not very specific for OCD. I should have a better answer, almost like sociopolitically, epidemiologically, why it is that OCD and depression seem to be the two disorders for which the highly invasive treatments got advanced the fastest. If you look at places like China and, there are people exploring, people move fairly quickly after that to psychotic illness and addiction. And, sort of from the epidemiologic perspective of like total disease burden through the world, those are kind of the big three. But so at any rate, I think in answer to your question, right now, DBS is not one thing. DBS is a mode of intervention. And then hopefully, if my talk was successful in any way, it's that circuitry matters. And as is true with the history of pharmacotherapy and psychiatry, a lot of what we've gotten to serendipitously and then try to rationalize after the fact why that thing worked for this. I do believe that with where we are headed with brain imaging, there is the potential to identify, to, in essence, create circuitry knowledge that then can be leveraged for more likely to be affected, targeted neuromodulation. And that has guided, TMS is probably a better example where, and now that people have deep TMS methods, there are opportunities to sort of, we have hypotheses that should be tested empirically by trying these treatments. But it is different targets. Area 25, I think, Helen Mayberg's belief is that if it works in depression, it's because it gets to area 25 directly or indirectly. It's different circuitry.
Yes. Back in the back. Yep.
Hi, wonderful talk. I'm Xiaofu. I'm working with Dr. Richard Marsh and also Brad Simpson on the OCD and specifically focus on the DTI findings there. So I have a question regarding the therapy. You have many target regions, right? So those target regions is based on the population level findings. Instead of, I'm wondering, for example, if you target the coded or ACC, is that based on population level findings? So my question would be, is it possible based on individual based? Because we don't know which region will be the target specifically for one specific patient, right? We don't know which region where it works or not.
Yes, yes. I really appreciate that question. You let me know if I'm not really asking the question that you intended to have me answer. But most of the data that we're showing are group data. And the question becomes sort of at the individual level, is that really the right answer? And how do you really move to think about even personalizing? And so I would just say, again, whether it's TMS or DBS, there is actually a fair tradition in neurosurgery to very much think about personalizing treatment so that it's not just, it's not a set of TALRAC coordinates or MNI coordinates, based on 100 people studied elsewhere, that there's something you're having this actual patient do that guides you to precisely the right target. It also gives me an opportunity to speak to one phenomenon that I know, given the time and the talk, I didn't, which is that, we certainly know well that OCD is not a monolithic, entity. There's huge heterogeneity clinically in what is OCD. And a lot of smart investigators have explored different ways to sort of parse up the population of people who have OCD. The early tradition was in subtypes, and I think that ended up being probably less fruitful than what we've now moved to, which I think is more of a dimensional approach. And the reason that the dimensional approach is more helpful is that often people with OCD have a range of different symptoms. And just because they have one set of symptoms doesn't mean they don't have the others, but they have the various types of symptoms in varying proportions. And that sort of defines the uniqueness of each patient. So I totally agree with you that as one moves translationally to newer, better treatments, a piece of that has to be tailoring the treatment to the patient and to fully respect the heterogeneity of clinical presentation and hence underlying pathophysiology across the population of people who are said to have the disorder. So I hope that responded to your question.
Yes, thank you.
So thank you. I think we're out of time. So let's thank Scott and Amit again for their Glassman Award and lecture. And there's lunch with Scott up in the multipurpose room on the 6th floor. You know this, but the residents go, and then you have more meetings.
I think more.
Yes, she definitely.
I don't really know, so that'll be great. She's on
Metro, yes, and I think she not only is she going to collaborate on the OCD work, but I think she would really value your input in transition. Yes.
Hey, there she is. How you doing?
I'm so busy. I don't get a chance to see you at all today, so I just want to give you a hug and say hi. And well, great talk. It was nice. Thank you. For sure. Yeah. The whole career.
I asked about you last night with the people I was with last evening.
Oh, yeah? Oh, well,
I was
that's
running
sweet.
down all the people who I think I was